Hi, everybody. What we're going to do is quickly review as many of the different types of signaling pathways in cells as we can. And I'm going to do this on a whiteboard. So if we switch to that, I'll show you what we have here, which is a cell with a cell membrane out here, a nuclear envelope, double membrane, and then a chromosome here with one particular gene exposed. And what we've talked about in the past are different types of signaling pathways that can exist in cells. The first one we talked about are receptor tyrosine kinase pathways, and that's what's described here. A receptor tyrosine kinase is a receptor that impairs if it binds to its peptide hormone. We've got two of them here. It autoactivates and acts as a kinase. And if you don't remember what a kinase is a molecule, is a protein that can add phosphates to other proteins. So a protein kinase, and hopefully this will there we go, see if we can get some focus. This RTK, this kinase that is a receptor, so that's RTK. It is a receptor that is a kinase, means it adds phosphates to tyrosines on proteins. So the RTK is going to find a target protein and convert it from one form to another. It's going to take its target protein and add a phosphate to it. And that's going to change its shape, which is going to either activate or inactivate it. For simplicity's sake, we're going to say that this is now the active version. Remember that phosphate comes from ATP. So in doing so, the ATP is, used up. A phosphate is transferred from the ATP to this blue molecule. And what we talked about last time as well is that the targets oftentimes end up being kinases, leading to what's known as a kinase cascade, which ends up in multiple steps, eventually leading to some sort of target. Now that target can be many things. Target could be a protein that triggers exocytosis of, maybe a hormone or an enzyme. It could lead to activation of an enzyme. That's a possibility. This pathway could lead to movement or cellular contraction if it's muscle. But what we really ended up talking about last time is the most long-lasting, which is to eventually activate a transcription factor. In this case, we described it by being active when it's phosphorylated, when a phosphate's been added to it. And that transcription factor enters the nucleus. It is allowed to pass through the nuclear pores. And it finds its way to the beginning of a gene, an area called promoter, where it binds to that beginning of the gene and recruits in the appropriate polymerase that reads that gene and causes that gene to be transcribed, later translated, leading to a production of a protein. So what I've described here is one type of pathway. And so this is an RTK pathway, and while it typically leads to enzyme activation in many cases, I'm describing it as activating a gene. All right, so this is one type of pathway, that is the RTK pathway. And in many cells, that's used for responding to, say, growth hormones. Right? So maybe this is how you turn on the genes to cause growth or division. The other type of pathway that we discussed briefly were steroid hormones, which pass directly through the membrane, which is why steroid hormone receptors tend to be found in the cytosol or the nucleus. And those receptors double dip. Those receptors are transcription factors. They are directly activated by steroid hormones, not peptide hormones. Okay, but what this video is about are another type of pathway entirely. So these are going to be the most common types of pathways in your cells by far, and these are called GPCR pathways. And so GPCR is an acronym. G stands for GTPase, P stands for protein, C stands for coupled, R stands for receptor. So these are GPCR pathways, and here is the first G protein. Now, before we get too far, I just want to remind you, it's basically going to end up with the same results. We can either activate an enzyme, trigger exocytosis or endocytosis, trigger movement, trigger cell death pathways, or more commonly, activate a transcription factor. Now, here's what I want to just delineate here. What we talked about is the signal transduction pathway going through the cytosol. Over here was a kinase cascade. And the way the kinases were activated were by phosphorylation, by adding a phosphate. What I'm going to quickly just show you here is that not all kinases are activated by phosphorylation. Maybe in this case, you might have a kinase over here, and I'll draw it as square, because I guess that's how I drew the kinase over there. This kinase might be activated by binding to something called cyclic AMP. We'll talk about what that is. And another kinase, a different kinase over here, might be activated by a different molecule that changes its shape. Remember, we just have like switches. So kinase off, kinase on. Kinase off when it doesn't have cyclic AMP, kinase on when it does. Kinase off when this is by itself. This kinase is on when it's bound to a calcium ion. And in the end, again, the kinase can activate a transcription factor, go into the nucleus, activate genes. So in the end, we're still talking about the same end targets. This kinase or this kinase might activate exocytosis or an enzyme or movement or lead to a transcription factor, which goes into the nucleus. All of these are the same. So how do you get these kinases active? That's where we're going to connect to the G proteins. So I'm going to draw two different versions. So I'm going to draw two different G proteins, one that leads to this, one that leads to this. And I'm warning you a little bit that these tend to be pretty complex. I'll try my best to simplify this process, but actually, draw two G proteins as far spaced as I can. So these are membrane proteins. And of course, they have a binding site for a hormone. And so this is going to be a different hormone out here. And this is going to be a different hormone here. The hormones, when they bind the receptors, obviously cause those proteins to change shape. Now, what makes them special is the G protein coupled receptors are coupled to things called G proteins. And that is really the focus of today's, I guess, lesson, if you want to call it that, is what a G protein is. And G protein is, again, coupled, like interacting with the receptor. So I'll draw it here. And they have on and off switches as well. Just like this receptor is off when there's no hormone, the receptor is on when it has a hormone. This kinase is off when there's no calcium, it's on when there is calcium. This kinase is off when it doesn't have a phosphate covalently bound to it. It is on when it has a phosphate bound to it. These G proteins are the same. They are off when they are bound to GDP. Guanosine. So this is just like adenosine diphosphate, just with guanosine. Okay. And they're on if they're bound to GTP. But these don't have any GTP. So the way these pathways work is that when a hormone binds to this receptor here, it changes shape, it undergoes a conformational change, allowing this G protein to dump its GDP and instead pick up a GTP. I'll state that again. This G protein, which is holding a non-useful GDP, is able to drop it and pick up a GTP. Now, this G protein is active. It is bound to a GTP. So a GTP comes in, the GDP is dropped, we have an active G protein. Same thing over here. GTP is allowed to exchange with the GDP, And now this G protein over here is activated. So everything has an on and off switch. It was off, now it's on. So what do these proteins do? Well, these two proteins, these G proteins, are able to activate the next step in the pathway, which is not a kinase directly. It's going to be something that leads to either cyclic AMP or calcium increasing inside the cell. And so I'm going to draw you two enzymes or proteins that are found in the membrane here. And here are two possibilities. One is an enzyme called adenylyl cyclase. And the other, which those I should draw in a different color, but I'm not going to because I've run out, is a calcium channel. And so what I'm telling you here is that the G proteins, when they're on, they activate these enzymes. Give myself a little bit of spacing here. And so when these enzymes are active, here we go, they have a particular role. This is the adenylyl cyclase. Adenylyl, I don't even know. And this is a calcium channel. When these two proteins are activated, they lead to these molecules becoming higher in concentration. So adenylyl cyclase is going to convert something into cyclic AMP. And when there's cyclic AMP around, that is what converts the inactive kinase to the active one. This calcium channel allows calcium in, either from the ER or from the extracellular fluid. And that calcium converts the inactive kinase into the active kinase. Now, what are all these for? Now, I just want to take a step back and remind you that the whole purpose of this is to do certain cell activities. This pathway might activate the result, cell growth, or in cell division. That might be what this hormone that binds to the RTK of that cell does. This over here, this G protein coupled receptor pathway, might activate specific genes that lead to a different activity. This pathway might lead to a different activity. So when you think about any particular cell type, usually there are 3 or 4 or 5 things that that cell can do. It can grow and divide, maybe this pathway. Maybe it can do its superpower, exocytose a hormone. Maybe that's this hormone. Maybe it's this pathway here, leading to exocytosis. Maybe that cell can do something different. Maybe it can move. Maybe that's this pathway. And there might be other pathways as well to lead to cell death or differentiation. Maybe this pathway over here actually semi-permanently turns on or off genes. So this becomes a more specific cell type. But that's the pathway here. These are the different pathways in. Just to reiterate this pathway over here with the G proteins, hormone binds the receptor. The receptor allows the inactive G protein to get rid of its useless GDP and pick up a GTP. That's now on. Again, receptor, when a hormone is bound, allows the G protein it's coupled to get rid of its GDP and pick up a GTP. That G protein is now on. The purpose of G proteins is to activate enzymes, whether it's adenylyl cyclase or guanylyl cyclase, which is for G, or this calcium channel here to allow for calcium influx. That activates typically kinase. That activates kinases typically, which then lead to the end results. That's it for now. That already took too long. Feel free to rewatch any particular part that you were unsure about. I'll see you guys next time. Bye. One quick addendum. When we think about amplification in this process, it was pretty easy to understand, or relatively easy with the kinases, in that one kinase could activate 20 of the next. These are enzymes, so it's possible to imagine that this particular kinase here, the blue square, might be able to activate, say, 50 of the transcription factor or 20 of another molecule. And that's where we get the amplification here primarily. The amplification is less obvious right here because the hormone binds directly to one receptor. The receptor binds to one G protein. The receptor binds to one G protein. The G protein binds to one enzyme. So where is the amplification? Well, there are the kinase steps that are going to happen down here. But I want you to know that this is also amplification right here. We're going from one enzyme that is active to tons of cyclic AMP being produced. This is an enzymatic reaction that takes ATP and converts it to cyclic AMP, which is just being used as a signaling molecule. Same thing here. We have one hormone binding to one receptor, activating one G protein, activating one channel, which leads to thousands or tens of thousands of calcium ions entering. So most of this is 1 to 1 to 1 to 1 to 1, but then we get massive amounts of cyclic AMP or calcium entering the cell, activating massive numbers of, in this case, kinases. And you can see there are three different types of kinases, those that are activated by phosphate groups, right, when they're covalently bound, those that are activated by calcium ions, and those that are activated by cyclic AMP. For simplicity's sake, I tend to think of these as A kinases, cyclic AMP. I think of these as B kinases, and I think of these as C kinases. So protein kinases, A's, B's, and C's, C for calcium in this case. That's the overview. I will see you guys next time.